In this video, we will conclude our discussion of what influences genetic diversity, specifically focusing on natural selection. And natural selection is different because in the previous two videos, we've talked about processes that lead to non-adaptive change. So they're random chances and they're generally decreasing genetic diversity. Natural selection is different because it's adaptive and it can also lead to either increases or decreases in genetic diversity depending on the type of selection, and we'll talk about that. And remember, we said in a couple videos ago that we're talking about these different factors of drift and selection, and they're not acting in isolation. They can be interacting with each other. And part of the factor that determines which one of these influences is going to have a stronger effect in a population is the effective population size. So if we have these two different general types of populations, if we have a small effective population size, in general, drift is going to have a really strong effect, which we talked about. Drift has a larger effect in small populations, and then selection will be a smaller effect. In contrast, if we have a large population, that means that selection is going to be able to have a larger influence, and conversely, drift is going to have a smaller effect. So that's really important to think about in terms of your effective population size, which process, either that adaptive process of selection or that random process of drift, might be more influential. Okay, so here, this is an image showing the different types of selection. So we'll walk through this type by type, but first let's go over the basics of what this graph is looking at. So in the first row, we have a parental population. These curves are phenotype curves. So essentially the different phenotypes are here on the x-axis, and the y-axis represents the frequency. So here, originally for this directional selection example in the parental population, we have a normal distribution of different types of phenotypes. The red X's indicate individuals in the parental population that are not going to survive to produce offspring, so those particular phenotypes are going to be lost from the pool, resulting in offspring, which are shown down here with shifted phenotype curves. So in this case for directional selection, because we got rid of the phenotypes on the left side of the curve, what we did is shift that to the right, and now we have a phenotype curve for the offspring that is directionally shifted, hence the term directional selection. But that's the way to read these different images shown here. So if we start with directional selection, the effect of directional selection is to decrease genetic diversity. I'll abbreviate that by GD. So what happens here is you're essentially shifting your phenotype curve to one particular direction, one particular edge of the phenotype range. An example of this is with sticklebacks. When stickleback fish, for instance, move from salt to fresh water, they experience a reduction in the number of armored plates that are on their side. So that's what's shown in the red here. That's an example of directional selection because you're shifting that phenotype in one direction, in this case, a smaller number of armored plates. For stabilizing selection, if we look at these red regions here, we're getting rid of the ends, and what we're doing is instead emphasizing this middle portion of the phenotype range. So stabilizing selection also reduces genetic diversity because you're specifically selecting for the individuals here in the middle. The example of this that I teased at the end of last module, that is one of my favorite things, it's super interesting, is called counter-illumination camouflage. So the idea is that many individuals that live in the deep ocean where it's completely dark, if they are swimming along. Once they pass the sun, they're going to end up blocking out the sun and causing a shadow on their underside. If there is a predator below them or a prey, that change in light is going to be visible, and that other organism will be able to notice them from that change in light. So a lot of organisms like squid are shown here. They've developed something called counterluminance camouflage, where on the bottom they have these symbiotic bacteria that actually produce their own light. And so when you then pass across whatever light source, the sun, they're not producing a shadow anymore because they're producing their own light. That's super cool, right? And the reason why this is an example of stabilizing selection is that if you have too much light, then you're going to attract attention to yourself, right? Then you're like a beacon. But if you have no light, then you're casting that shadow. So I like to think of this as kind of the Goldilocks type of selection, where you want it just right in the middle, where in this example of counter-illumination camouflage, you're producing just enough light to make yourself not noticeable. All right. And then the last type of selection, which is also one of my favorites and is super cool in terms of examples, is disruptive selection. And here what we're doing is eliminating this middle part, or decreasing phenotypes in the middle, and instead we're emphasizing the two phenotypes on the ends. So this type of selection is actually increasing our genetic diversity because we're maintaining two different ends of the phenotype spectrum. One of the examples here that I'm showing is with cuttlefish. So an example of disruptive selection is the maintenance of two different types of males in a reproductive system. So you have the males who try to mate with females by being dominant and being impressive, and then you also have sneaker males who use different tactics. So here in this example, this one right here is a male. The one on the left is a female. And cuttlefish do this super cool thing where they communicate to each other through changing color patterns on their bodies. And they have different patterns that mean different things. So this sneaker male here in the middle, he's showing different things to the female on his one side and his rival male that he's trying to trick on his other side. So the whole strategy for sneaker males is basically to sneak in and mate with the female before any of the other males notice. So what this guy is doing is, to the female, he's showing a mating signal. He's showing these different stripe patterns. He's trying to attract her to mate with him. To the male on the other side, he's doing quote unquote business as normal. He's pretending to be a female, essentially. So this is, again, I think super, super interesting. And it's an example of disruptive selection because we're maintaining two different types of males in this system, the sneaker males and also the more traditional dominant males. Another type of selection that can help to maintain genetic diversity is called balancing selection. And I have a teeter-totter here because the idea with balancing selection is that two different alleles, so you might have allele 1, allele 2, at different points in time or different points in space, the allele frequencies can either increase or decrease depending on the situation, but neither of them is completely eliminated from the population. So balancing selection is maintaining genetic diversity because it's maintaining different alleles. Even though one of them may be at a low frequency at one time point, it can shift to a higher frequency at another time point when it's more appropriate. An example where they have found this type of selection to be acting, this is the citation. Bay, she actually works at UC Davis, and they have looked at corals in Ofu Island, which is in American Samoa, and they study these corals in two different types of reef habitats. Depending on the particular location of the reef and how it's inundated by tides, some of those reefs are warmer than other ones. And what they found is that in these different habitats, balancing selection is actually selecting for different alleles based on the different habitats. So that's how this spatially varying selection comes into play. So selection is acting differently to maintain different alleles depending on the habitats and their corresponding temperatures. So what does that look like? This is one excerpt of a figure from Dr. Bay's paper, and I'll walk us through this. So in this figure here, these red and yellow colors, each row is a different SNP that she identified in one particular gene they found that had a lot of differences between the two different coral pool habitats. Each of the SNPs here indicated with an asterisk is one that causes an amino acid change. So remember we talked about that way back when. That's a non-synonymous SNP. Each row is a different coral colony, and it's divided into two general categories. This top category is a highly variable pool, meaning the temperatures are highly variable and it gets warmer here. And the moderately variable pool on the bottom is a little bit cooler. The temperatures aren't as extreme. And then the colors in this, it's called a heat map. The colors indicate the particular genotype for that coral colony at that particular SNP. So we have two different alleles, A and B, and then we also have our heterozygote. In general, at the warm site, this B allele is more common. It's more beneficial for helping these corals survive these really warm temperatures. But if we look in the slightly cooler, moderately variable pool, we still have some instances of that B allele. It's just not quite as common. So the argument that they made in this paper is that balancing selection is maintaining this allele at low frequency in these other populations, and that's cause for hope. I'll try to leave this on a positive note. I know some of the earlier parts of this module were a little bit sad in terms of conservation, but what this indicates is, as stated here in the text, this natural population has a reservoir of alleles, that big B allele, that's pre-adapted to high temperatures, suggesting that there is a potential for a future evolutionary response to climate change. So, and this also ties it all back to genetic diversity that we've talked about before. So again, we said higher genetic diversity is needed to be able to adapt to changing conditions, right? Evolution is only able to act on variation that is already present in the population. So if we think about this moderately variable pool, overall right now, most individuals have this A genotype. Some of them, though, do have this B allele. So if temperatures were to shift in that pool to become warmer, there is the potential for those individuals that have that B allele to be able to survive and then reproduce. And through selection, you could end up with a genotype pool that looks more like what's at this highly variable pool right now. So the idea of balancing selection is to be able to maintain these different alleles in different appropriate spatial habitats. But if that habitat changes, there's a potential for adaptation to be able to survive long term as the environment changes.